Now, eukaryotic cells are fundamentally different from prokaryotic cells in containing a nucleus, among other things. But the nucleus, for our purposes, is the most important consideration. It's in the nucleus, of course, where the DNA is kept, whereas prokaryotic cells don't have a nucleus. The DNA is floating around in the cytoplasm, so that transcription and translation in prokaryotic cells can both occur in the cytoplasm, whereas in eukaryotic cells, the transcription occurs in the nucleus, but translation occurs in the cytoplasm. So transcription and translation are separated from each other in eukaryotic cells. Another big consideration about eukaryotic cells is the size of the DNA of eukaryotic cells. As I mentioned in class, if we take all of the DNA of a human cell, for example, and we stretch it end to end, from one single cell, there's 7 feet of DNA. Well, the cell, of course, is smaller than your eye can see, and so it means that that DNA has got to be wrapped up tightly in order to fit into the confines of the nucleus. And by the way, the nucleus isn't much bigger than a prokaryotic cell. Okay? A prokaryotic cell has on the order of about, for E. coli, for example, about 6 million base pairs of DNA. It's circular. Okay? The eukaryotic genome for a human being, for example, is 7 billion base pairs in size. Well, that's about 1,000 times more DNA in the same volume as an E. coli cell has. So it means there's got to be some other considerations about packing that up. Well, one of those considerations is shown on the screen here, and that relates to the packing process that involves using proteins called histones around which the DNA coils, and we can see some of that coiling occurring here. The coiling of DNA around histones creates structures that we give various names to. The simplest structure is something called a core particle, which is simply the DNA wrapped around the histone. It goes a couple times around there are 8 histones in an individual one of these circles that you can see, and that forms something called a core particle. If we look at a core particle and then we look at the strand of DNA coming off of it, that creates a fundamental, or something we call a fundamental unit of this bigger structure, and that fundamental unit is called a nucleosome. So a nucleosome is a core particle, which is just the circular part, plus the associated strand of DNA coming out that we can see right here. If we take all of those nucleosomes and we put them all together, we make something we call chromatin, which is part of a chromosome. Okay? So we got several orders of structure. We have a core particle, the smallest individual unit. We have a nucleosome, which includes the extended sequences around it. We have chromatin, which is when we add all these together, and when we put all the chromatin together, we make the structure that we see in the microscope called a chromosome. Well, why do I talk about all this? Well, it turns out that, as you can see on the screen, there are some very big considerations for the way that the chromatin is organized. Okay? So if the chromatin is organized in a loose fashion, as you can see on the left, then that loosely organized chromatin is called euchromatin. And that form of chromatin is transcriptionally active, meaning that the RNA polymerase, and by the way, in eukaryotic cells, there are 3 RNA polymerases. The RNA polymerase can find the DNA, get into it, and start making messenger RNA off of it. So euchromatin is where we will see transcription actively occurring. The other kind of chromatin that we see is what's called heterochromatin. And you can see in this figure that heterochromatin is very tightly packed. The individual nucleosomes that are there are scrunched together very tightly, and a good deal of our DNA is commonly found in this form. And you could imagine that in this form, where the nucleosomes are very closely packed together, that the RNA polymerase would have a hard time finding the relevant sequences in the DNA, and so heterochromatin tends to be transcriptionally inactive, meaning that there's not much transcription going on. On the screen, we can see the DNA around the region of the transcriptional start for a eukaryotic cell. And this only actually shows part of the overall structure of the DNA, but I show it to help to bring about a bit of the idea of the complexity of the regulatory sequences of the DNA. The regulatory sequences, of course, being what I referred to earlier as the promoter in the case of the prokaryotic cells, but actually can extend in the case of eukaryotic cells for hundreds or even thousands of base pairs away from the transcriptional start for a eukaryotic cell. We have in the case of the eukaryotic cell, of course, the transcriptional start site, just like we had for a prokaryotic cell, but we have other considerations in terms of factors that, or sequences that are bound by factors that affect transcription. Now, these are important. Before I talk about the individual components, I want to talk about the importance of having many controls for genes in eukaryotic cells. When we look at a multicellular organism like myself, or a plant, or a dog, or some other animal, we recognize the fact that even though all of the cells of that organism had identical DNAs, the cells themselves were not identical. So my skin cells are different from my eye cells, or different from my bone cells, or different from my muscle cells, etc. And those individual cells have different needs in terms of which proteins they have to have made. So it's important that the cell be able to make the proteins that it needs and not waste energy making proteins that it doesn't need. Well, to be able to control that, that means you have to have multiple control regions, and that's in fact what eukaryotic genes have. So you see a very tiny part of that picture on this schematic of this gene here. Like prokaryotic cells, eukaryotic cells have a sequence near the transcriptional start site called a Pribno box that is rich in AT sequences, okay? That richness in AT sequences is called a Tata box, and it's very much like the TA rich region that we saw in front of prokaryotic genes. In the case of the eukaryotic cells, however, we see that the location of that Tata box is not fixed. It's not exactly at -10. In fact, it's most commonly found between about -30 and -100 of the location of the start sequence. Not all eukaryotic genes even have a Tata box, but all eukaryotic genes that have a strong promoter, that is, that they make a lot of messenger RNA, all of those eukaryotic genes will have a Tata box. We see that there are other sequences that are located 5', and in this case we're going 5' to the transcriptional start site, things called CAP boxes and GC boxes, and there are other boxes as well that tell the cell that this is first of all, it is a gene that we want to make, and the more of these individual sequences that we have out there, the more fine-tuning of that transcription can occur, meaning that some cells will have a protein that recognizes this sequence and say, okay, this is a gene that we need. So a skin cell might be able to recognize this sequence, for example, but a bone cell might not be able to recognize this same sequence and therefore wouldn't transcribe that particular gene. So the ability of cells to have many control regions allows them to have differentiation of function and differentiation of expression of proteins, and therefore differentiation in the appearance and the activities of the cells that contain them. The last thing I'll mention, and it's not shown on this figure here, is another unusual sequence that's found in DNA for eukaryotic cells, and that's a sequence that we call an enhancer. And enhancers are unusual in that they can be located hundreds or thousands of base pairs away from the transcriptional start site. Enhancers appear to help cells to do that differentiation function, as I noted. So enhancers are going to be bound by specific proteins, and those specific proteins are found only in certain differentiated type of cells. So a bone cell might have a protein to recognize a particular enhancer, whereas the skin cell might not have that. Again, so we could imagine that if the bone cell has that protein, it binds to that enhancer is going to help enhance and make that messenger RNA that's located thousands of base pairs away. Well, how does it do that? How does an enhancer help an RNA polymerase thousands of base pairs away from where it's located to stretch and to know where that transcriptional start site is? Well, it turns out that DNA, of course, is flexible. And so if you go far enough, DNA can actually loop around, okay? And something that's thousands of base pairs away, when it loops around, can be brought into close proximity, in this case, of a promoter. So if we could imagine up here where my finger is that we have a looped DNA that is bound to an enhancer, that enhancer could interact with the promoter here and facilitate the transcription that occurs downstream to make the desired messenger RNA. So enhancers are unusual sequences, but they do appear to play some role in eukaryotic cells. We don't really see good examples of enhancers in prokaryotic cells. Now this slide shows some features of a eukaryotic messenger RNA. And it's important to note that there is some processing that occurs for a eukaryotic messenger RNA that does not occur for a prokaryotic messenger RNA. So there are also some regions that we see in eukaryotic messenger RNAs that are not commonly found in prokaryotic messenger RNAs as well. So eukaryotic messenger RNAs tend to have what we call long untranslated regions, or UTRs. And those UTRs are located both in the 5' end of a given gene and in the 3' end of a gene. Right? Now these untranslated regions are, as the name would suggest, not translated. The ribosome doesn't make protein from them, but these regions may play roles in governing how much protein is made from a given messenger RNA. And that's really beyond the scope of this class. So I won't say more about that, but that's why you see the UTRs on here. Other sequences that you see on here are important to understand. And one of these sequences, you see two of them labeled here as introns and exons. And introns and exons are something that are unique to eukaryotic cells. They're not found in prokaryotic cells. What are they? Well, if you follow the progression, starting at the top and going down to the bottom, you'll notice that the introns that were at the top disappear, and the exons are what is left behind. Well, that tells us, of course, that introns are sequences within a messenger RNA. Remember, we're looking at messenger RNAs here that are removed, and they're removed in a process we call splicing. So splicing is a process that removes introns and joins exons to make what we call a mature messenger RNA. So that splicing process, I'll say a word about in just a minute, but the splicing process is something that occurs in eukaryotic cells, not in prokaryotic cells. There are other processing things that happen to eukaryotic messenger RNAs that don't happen to prokaryotic messenger RNAs that I want to say a little bit about. One of these is what you can see on the screen, and it occurs at the 5' end of a eukaryotic messenger RNA. This sequence at the 5', or this structure at the 5' end, it's not really a sequence, is known as a cap. So a eukaryotic messenger RNA is made to start very much like a prokaryotic messenger RNA. The RNA polymerase comes on, it starts the first nucleotide, it goes along through the process of elongation and termination and does its thing. Those other processing things like splicing that I talked about also occur to the eukaryotic messenger RNA. However, at the 5' end, after that transcription has started, there's a modification of the 5' end that occurs to put this cap structure that I'm talking about here. The cap structure you can see on the screen is shown in blue, and this cap structure is a modified form of a guanine nucleotide. Now, we don't need to concern ourselves in this class with the exact structure of that, but suffice it to say that that guanine nucleotide is joined to the 5' end, and here's the 5' end of the messenger RNA that you can see here. It's joined to the 5' end of the messenger RNA in a very unusual bond, and you can see that bond right there. It's a 5' to 5' bond between the modified guanine nucleotide and the 5' end of the messenger RNA. Now, this unusual structure, as I said, is called a cap, and it appears to play at least two functions. One function is to protect the 5' end from degradation by nucleases. Nucleases that start at ends, called exonucleases, cannot get past the cap. So basically the 5' end of the nucleotide, of the messenger RNA is completely protected from nuclease degradation. That turns out to be important for the stability of messenger RNAs. Okay, now that's one consideration. Another consideration is that in the process of translation, the ribosome actually looks for the cap and uses the cap as part of the process of initiation of translation in eukaryotic cells. And I'll have a lecture where I talk a little bit more about that later. So another modification that happens to eukaryotic messenger RNAs occurs at the 3' end of the eukaryotic messenger RNA. This involves the addition of hundreds of units of the nucleotide that contains A, okay? So it's called a poly-A sequence. And that poly-A sequence has hundreds of A residues at the 3' end. It would seem like that's a fairly mundane kind of a thing to do. Why does the cell do it? Well, again, there appear to be two reasons, and those two reasons are pretty much the same as what happens at the 5' end. First of all, the addition of hundreds of A's doesn't stop a nuclease from acting, but it certainly slows it down. So the longer the number of A's that are added to the 3' end of a eukaryotic messenger RNA, the longer it takes an exonuclease to chew through it and degrade the messenger RNA. So it's a protective function, kind of like the cap, although the cap is more protective than the poly-A itself is. The second function of the poly-A tail on a eukaryotic messenger RNA is in translation. And like the cap structure that I talked about before, the poly-A tail is recognized in the process of the initiation of translation. And again, as I said, I'll have a lecture later that shows that in a little bit more detail. You might wonder, why do cells want to degrade their messenger RNAs? Why do they want to have them around all the time? And the answer is that cells don't need a given protein being made all the time. So it's important not only that cells be able to control when to make the messenger RNA, but also to control when to make the protein. And so there's a variety of ways that the cells can control that. One of the most common ways is degrading the protein messenger RNA. If the messenger RNA is degraded, then of course a protein can't be made at all. Now, the other process that I talked about with respect to modification of messenger RNAs relates to the phenomenon called splicing. And splicing is something that has a lot of complexity and more than we're going to go into in this class, but I want to just say a few words about what you see on the screen here. So if you remember from what I said earlier, in the process of splicing, intron sequences within a messenger RNA, such as what is shown right here, intron sequences are removed and exon sequences on either side of it are joined to form a mature exon-joined messenger RNA, such as you see here. The process of splicing occurs in a structure we call a spliceosome. And that sounds like a ribosome, but ribosome is where translation occurs. A spliceosome is where splicing occurs. So don't confuse those two terms. What you see on the screen is a depiction of what's actually happening in the spliceosome. And I'm not, or spliceosome, they're the same thing. I'm not going to talk about all of the structures of a spliceosome or how this occurs, other than to point out a couple common things at the very top here. The first of these is if we look at the intron sequences for all of the individual introns that are known in eukaryotic cells, they have a couple of common features. One common feature is found at the 5' end of the intron. And this is a sequence that appears almost every single time, and it's known as a GU. That GU is an indication that this is a 5' end of an intron. If we look at the 3' end of the intron, there's another sequence that we almost always see, and it's AG. So the 5' end has a GU, the 3' end has an AG. About 30 nucleotides ahead of the AG, there is commonly a sequence of nucleotides that's very rich in pyrimidines, C's and T's. And in the middle of that, there's usually an A. And that's what you see happening on the screen here. So in the process of splicing, what actually is happening is the G that is at the 5' end of this intron is attacking the A that's found in that pyrimidine-rich sequence and forms a bond like you see here. In the rest of the process, the bond between the 3' end of the intron here and the exon is broken, and the two exons are joined together to make the structure that you see below. That releases the intron in the form that we call, as you can see over here, a lariat structure. And it's called a lariat structure because it has a 3' end sticking out like the end of the lariat, and it has a loop structure that you can see, I can't see my hand, but has a loop structure that contains a very unusual bond. The bond between the G and the A that I talked about earlier is a 2' to 5' bond. It's the only place we see such a bond occurring in molecular biology, and it's what holds the lariat together. The lariat, for the most part, is simply degraded and not used for anything else, although there are a few exceptions to that. For our purposes, we'll just consider it to be degraded. Now, people ask, what's the advantage of splicing? Why do eukaryotes go to the trouble of making a messenger RNA that contains sequences that it later removes? What good does that serve? Well, this figure illustrates that to a limited extent. One of the advantages that we know of splicing is that cells can splice things in different ways. And when they splice things in different ways, they can make different forms of a protein. So remember, of course, that we are differentiated. We have differentiated cells, the bone cell being different from a muscle cell, etc. And so we talked at one level about needing different genes. So if I need a protein in a bone cell and I don't need that protein in a muscle cell, then I have ways of controlling, for example, its transcription to make it in a bone cell, but not transcribe and not make it in a muscle cell, all right? Well, sometimes the same protein is needed, but in a slightly modified form. So if the cell is able to make a messenger RNA, such as you see here, and splice it in one way so that the exons necessary for a protein's function in the bone cell are present, all right, then the bone cell will splice it in one way, but a different set of exons that's slightly modified may be needed in a let's say, a liver cell, and it will have its own way of splicing that so that it puts those exons together in a slightly different fashion and makes a slightly different protein. So the point of this is that splicing appears to allow a cell to increase the diversity of proteins that it makes given a specific sequence. So to give you an idea about this, human beings have the coding for about 20,000 different proteins. But from that 20,000 different proteins, we know that they can make at least about 100,000 different forms of those proteins, meaning that many proteins are made in multiple forms, and the way in which that occurs is by differentially splicing the messenger RNAs in different differentiated cells.